Module 6, DNA, Proteins, and the Cell Cycle. Mitosis. Last section we covered interphase of the cell cycle. I had mentioned that there are two major cell cycle phases, interphase and the mitotic phase. The mitotic phase is the part of the cell cycle when the cell actually divides in two. Both mitosis and cytokinesis happen during the mitotic phase. Cytokinesis is the process usually following mitosis or meiosis in which the cytoplasm of a cell is divided in two. Now, mitosis is when the genetic material and the nucleus divide. Cytokinesis is the process where the cytoplasm divides, splitting up the organelles and forming two daughter cells. We can break down the mitotic phase into 4 stages plus cytokinesis. They happen continuously, but there are specific things that occur in each step. The stages are prophase, P, metaphase, M, anaphase, A, and telophase, T, then cytokinesis. Some people remember the order of the phases by the acronym PMAT, P-M-A-T. Let's look at prophase first. Pro means forward, so you can think of this as the most forward or first stage. In prophase, the duplicated chromosomes coil up into their condensed club-shaped form, attached to each other by their centromere. Remember that in the synthesis stage of interphase, the DNA strands were duplicated. In prophase, the chromosomes become tightly packed and are visible under the microscope. They look like little X's. In interphase, you cannot see them very well because they're uncoiled chromatin. Also, in prophase, each set of centrosomes will migrate to one end of the cell. Centrosomes have centrioles that help to organize the centrosomes and help with cell division. The centrosomes extend microtubules outward and form a structure called an aster. This word means star-like, and the extended tubules resemble a star. As the centrosomes migrate to opposite cell ends and the microtubules from the aster grow and form spindle fibers, they form the mitotic spindle. Also at this time, the nuclear membrane breaks apart and exposes the chromosomes to the rest of the cell. Microtubules from each centrosome will attach to the chromosomes at their centromeres. Then, the spindle fibers start to move the chromosomes toward the center of the cell. This part is sometimes called prometaphase because it occurs at the transition between prophase and metaphase. The next phase is metaphase. You can think of it as the second or middle step because the prefix meta means after. It comes after prophase and M is for middle, so it's one of the middle steps. In this phase, the fibers have lined up the chromosomes in a row along a line that's called the equatorial plane or metaphase plate. This imaginary line runs along the center of the cell like the Earth's equator, and it is an equal distance apart from each of the two centrioles. This short phase just occurs when the chromosomes are lined up like this. Now, anaphase is the next phase, and this is when the microtubules on the spindle pull the chromosomes toward the centrioles. Because each sister chromatid has a microtubule pulling on it from the opposite direction, the sister chromatids are pulled apart and move to opposite sides of the cell. The prefix ana means apart, and the chromatids are being pulled apart. That means the chromosomes are no longer doubled and don't have that X shape. During this brief point of anaphase, the cell has doubled the number of chromosomes that it needs. Once the chromosomes are pulled apart from each other, the final phase of mitosis, telophase, occurs, and cytokinesis starts too. Telophase and cytokinesis can happen at the same time. The spindle starts to disintegrate because it has completed its job of pulling the chromosomes apart. The plasma membrane starts to pinch inward along the equatorial plane, eventually forming two cells. A nuclear membrane forms around each chromosome group in each cell, and the chromosomes start to uncoil from their supercondensed form, forming chromatin again. Now there are two daughter cells instead of one parent. In plant cells, cytokinesis is a bit different only because the plasma membrane can't just pinch off due to the cell wall surrounding it. So once the chromosomes are pulled to the centrioles on opposite sides of a plant cell, vesicles that have the building blocks for the plasma membrane start to line up along the equatorial plane and form a cell plate. The vesicles fuse together and form a new plasma membrane. Then, cellulose starts to form in the middle and forms a new cell wall that splits the plasma membrane into two. A middle lamella is formed, creating two cell walls. 2 plasma membranes, and 2 cells. You can see the stages of mitosis and cytokinesis here in this photo. Interphase is the time between mitosis events. Prophase is when the chromatin condenses into tight, more visible chromosomes. Metaphase is when the chromosomes line up along the cell's middle, the equatorial plane. Anaphase is when the sister chromatids are being pulled apart. And telophase is when they are fully separated and the cell is ready to pinch in two. It's not as easy to identify as the diagrams I used, but it gives you a good idea of what things look like in real life. Module 6, DNA, Proteins, and the Cell Cycle. Experiment 6.2, Mitosis. This is a great experiment because you're going to get to see cells that are actually undergoing mitosis. And so not only are you going to learn more about what those stages of mitosis look like, you can identify the stages of mitosis from animal cells and plant cells. So we have 2 prepared slides we're going to look at. One prepared slide is of an animal cell and one is of plant cells. The allium slide is from a root tip, and it's going to either say onion root tip or allium. Allium is the genus name. And the other one is the animal cell Ascaris mitosis. We're going to start with the Ascaris mitosis, and we're going to stick it under the microscope, put it on the stage here. I'm going to turn on the slide. There it is. Center it. All right, get it under good, decent magnification. Go ahead and make an illustration of what you're seeing there. And then we're going to move to a 100 power magnification. Our goal is to see condensed chromosomes. That when the chromosomes are condensed, that is an indication that the cells are undergoing mitosis. So when you see darker purple spots, at least in this example, I'm going to move it so I can get the field of vision around a bunch of them, because I want to be able to identify the various stages of mitosis that they're in. And then I need to go one more power of magnification up to the 400 power. And let's get closer here. See if I can adjust that light. It's really difficult to see. Let's see here. All right. And so using my fine focus, the goal here is to identify the various stages of mitosis: prophase, metaphase, anaphase, and telophase. And you can use figure 618 in your text as a guide to identify those various stages, and you'll just illustrate what you're looking at under the microscope at 400 power magnification to show those. Remember, metaphase is when they line up along the middle, so you will see them all lined up. Let's see if I can find some in there. Magnification. I actually found the 2 that have, it looks like a figure 8 or the duplicate nuclei, those nuclei where they're separating apart. That's actually the cells getting ready to divide at the end of telophase. When you see cells just start to condense, here's one where it looks like they're lined up along the middle. Let's see if I can put that to the center. That's metaphase. If you see a cell with no condensed chromosomes, that's interphase. That's the phase between. Inter means between mitosis. So some of these cells are not undergoing mitosis, and that would be cells that are in interphase. Okay, so let's go on. I think that's all we're going to be able to see with this one. Let's go and take a look at the plant cells. I'm going to turn it back to 40 power magnification. Remove my slide and then get my allium onion root tip. With the onion roots will grow from the tip outward. And so it's important to look at the curved tip portion of your specimen. So I'm going to focus. All of the mitosis will be happening towards the tip, because as that root grows down, that's where the growth is occurring, as the root grows longer and longer. So let's center that before we move up to the next power of magnification. Okay. And here we go to 100 power magnification. There we go. Seeing lots of dots, which is a good thing. Let's re-center it. Again, towards the tip, the curved tip. Each one of those dots represents a nucleus that's been stained. So now we can go even further in magnification. 400 power magnification. Again, very difficult to see. All right, I'm going to move towards the tip. Whoop. Okay, and it's very, very difficult to see at this power of magnification what's going on with those various nuclei. One thing that's different between the plant and the animal cells is that with the plant cells under mitosis, their telophase is a little bit different in the plant cells. Two nuclei that have split apart, but the cell has not divided yet. Because remember, plant cells have cell walls, and it takes more time for the cell to produce the cell wall and the cell membrane to separate the two new cells, that one cell into two cells. So when you see two nuclei together in a cell that is not yet split completely, that is indicative of it being in telophase. However, it is very difficult to see with the light that we have here. Hopefully you will get a much better image without having to go through the major process we have of camera to identify. And again, use the illustration that you have in your textbook that shows all of the stages of prophase, metaphase, anaphase, and telophase. And again, if the chromosomes are not condensed, then the cell is not in mitosis, it's in interphase. Let's revisit this diagram again and talk about the checkpoints marked on it. Normal cells will grow to a specific point, and then they will divide. To enter the mitotic phase, the cells have to have two sets of DNA plus all the organelles needed for two cells to survive. So if there are not enough organelles, then one or both daughter cells won't survive. Also, if there is damaged DNA, then that damaged DNA will be passed on from the damaged parent cell to the daughter cells. So this is where the checkpoints come in. You see, biologists learned that the cell uses internal checkpoints to make sure the cell is ready to divide. The G1 checkpoint is where cells have to meet the requirements for cell division. This means the cells have to have an adequate size, a good amount of ATP, and no DNA damage. Then, if everything is checked out to be good, the cell will enter the next stage, the S stage, where DNA is replicated. If anything is wrong with the cell will stop itself from moving on in the cell cycle, first trying to fix things in an extended G1 phase, or if not, it will enter the G0 phase and never divide. The G2 checkpoint is when the cell checks itself to make sure the newly duplicated DNA is correct. If the checkpoint mechanisms find any problems, the cell will be stopped from entering the mitotic phase. But if the new DNA checks out, then the cell enters mitosis. Now, you should not be surprised to learn that this process is much, much more complex than what I just described. But the fact that our cells have not one, but two complex checkpoint processes that help to ensure things in our cells are properly run is more testimony to God's amazing creation. On rare occasion, the checkpoints stop working, and cell reproduction can happen non-stop. This is what occurs with cancer. Cancer cells undergo uncontrolled division, which produces masses of cells that are called tumors. This is what cancer researchers focus on in trying to figure out how to stop these cells. Because cell cycle checkpoints are broken down in cancer cells, that's why they divide much more frequently than normal cells. Many treatments, such as radiation and chemotherapy, help to target those faster reproducing cells, which has created some successful treatments.